Hi everyone, and welcome to Biochemistry Practical Course 2. Today, we will be introducing you to SDS page, and we will virtually practice how to make an SDS page gel. The word SDS page is an acronym for sodium dodecyl sulfate polyacrylamide gel electrophoresis. This means that we will make a gel that contains sodium dodecyl sulfate together with polyacrylamide for electrophoresis. Electrophoresis means migration by electricity. We use this technique to separate proteins based on their molecular weight by running negatively charged proteins through an electric field in the porous gel made of polyacrylamide. Depending on the pH, every amino acid has a specific charge. This charge can be positive, negative, or zero. How does this happen? At low pH, both the carboxyl group and the amine group of any amino acid will be protonated. As such, the amino acid will be overall positively charged. Whereas, at high pH environment, both the carboxyl group and the amine group of any amino acid will be deprotonated. As such, the amino acid becomes negatively charged. At neutral pH, the amino acid is in a zwitterionic form. Zwitter is a German word for hermaphrodite. So, at neutral pH, an amino acid is considered an hermaphrodite. It has both negative and positive charges. Then, the overall charge of the amino acid at neutral pH will be zero. This principle is used during SDS page. Before the sample is loaded into the gel for electrophoresis, it is given a compulsory negative charge by adding negative compounds such as the sodium dodecyl sulfate, known as SDS, to the protein. Let's look at the basic principles of SDS page. The gel will be placed in a closed compartment which will be negatively charged. That is the cathode. Outside and beneath the compartment will be positively charged. And so, that's the anode. The protein will be negatively charged and it will be loaded at the cathode end. When electricity is applied, the protein will migrate towards the anode. As this happens, the protein will be separated by its molecular weight into several proteins that are present in the mixture as shown on this right side. So you have a separation of a mixture of proteins according to their weights. This is a cartoon image on the right side of this slide. On the extreme right side is a picture of a real SDS page gel image. To continue in the SDS page principle, let's look at the setup. The SDS page, it's a gel that is about 2 millimeter thick and it's set up within 2 glass plates vertically in the buffer. It's about 7 centimeter in length from top to bottom and it has 2 layers of different pH. The topmost part around here, it's about 1 centimeter layer. It's called the stacking gel and it has a pH of 6.8. And the lower part around here of the gel is called the running gel. It's about 6 centimeter in length from top to bottom. So you have 7 centimeter in total, 1 centimeter topmost part which is called the stacking, and 6 centimeter of the running gel, which is pH of 8.8. So you have 2 different pH setup, 6.8 at the top and 8.8 below it. Once a negatively charged protein is loaded here, the protein moves from cathode towards the anode in this direction. And the protein pH is far above 7, which is a sharp contrast to the pH of the stacking gel, which is 6.8. This sharp difference in pH between the protein and the stacking gel creates a strong ionic pull of the protein into the porous gel, and this will allow the protein to migrate for about 1 centimeter before hitting this lower environment, which is about 8.8 in pH. In this environment, there is no longer any ionic pull for migration of protein. Hence, the proteins will migrate according to their weight. The lighter protein will migrate faster, and the heavier proteins will drag behind, causing a smooth separation of proteins by weight. Now let's look at the components of SDS page. To make an SDS gel, we will need reagents. First, we need 30% acrylamide and bis-acrylamide. These serve as the matrix and the cross-linker to make a mesh of a porous gel. Secondly, we need tris buffer. We need 1.5 molar tris hydrochloric acid, which is at a pH of 8.8, and 1 molar tris that will be at a pH of 6.8. These are salts that will help electrons to flow. Third, we need 10% SDS. SDS is a negatively charged detergent. We also need catalysts such as 10% ammonium persulfate, which is the first catalyst and is an electron donor. We need TEMED, which is the second catalyst, and this is dimethyl aminoethane, shortened as TEMED, as the second catalyst. Now, how does this work? Polyacrylamide gels are created by the polymerization of acrylamide monomers with the methylene bis-acrylamide cross-linker. These are the acrylamide monomers. They form a horizontal chain that is vertically linked by the methylene bis-acrylamide cross-linkers. Thereby, they form a perfect mesh. The acrylamide monomers run horizontally and the bis-acrylamide run vertically, forming a perfect mesh. The higher the concentration of our monomers and our linkers, then the thicker the mesh of gel that is formed. And this whole reaction is catalyzed by APS and TEMED to form a very strong exothermic reaction. Before you continue into the material and methods, as a way of practicing biochemical calculations, using the formula of C1V1 is equal to C2V2. Here is a table of a number of reagents to be used to make SDSP gel. So, calculate the amount of the stock reagent that you need. The stock reagent is on this left side given to you, and you need to calculate the amount that you need to take to make the final reagent of 10 ml of gel. The final concentration should be in the middle, is the one in the middle, 12% weight by volume of acrylamide from a stock of 30% of acrylamide, 25% of tris from a stock that is 100%, and 10% SDS is the stock. You need to make 0.1% SDS as final concentration. So, and so on. Make this volume to appear on the red table. Good luck in the materials and methods.